Welcome to A Moment of Science, Please. My name is Hawwa.
And my name is Mohammad.
And we'd like to welcome Dr. Levy-Strumpf, who is an assistant professor in the teaching stream of the Human Biology Department and has done research in the field of genetics. Welcome, Dr. Levy-Strumpf.
Hi, I'm very happy to be with you today.
Thank you. So if you could please introduce yourself and tell our audience a little bit more about you and your background in education and what really interested you about the field of genetics.
Okay, so I did my PhD at the Weizmann Institute of Science, and I was studying cell growth control and apoptosis, which is programmed cell death. And I was really very interested in the process of metastasis, where cells can detach from their tumor, from the original primary tumor, and they acquire the ability to breach barriers, migrate elsewhere in the body, and generate secondary tumors. And very frequently in that process, the cells would acquire the ability to change their polarity. So I was very interested in polarity changes. And so I decided then, because back then I was working mainly with tissue culture, I really wanted to work in vivo. And I decided to choose a simple genetic system so I could ask more complex questions. So then I was contemplating between Drosophila and C. elegans and landed on C. elegans because it was the best system that was most suitable for the questions I wanted to ask. And so the reason why I gravitated to taking a genetic approach is because it allows you to carry out genetic screens. And when you carry out genetic screens, you can identify new genes that are involved in the process that you're interested in, and you can do that in an unbiased manner. So you don't know what you're going to discover, you're not biased in any way, and that can lead to breakthroughs. So this is how I landed on C. elegans. Did I miss anything? Let me see. Yeah, that's basically why I changed directions and I landed on C. elegans.
Perfect. So your past research includes studying the interconnection between the Wnt and netrin signaling pathways to orchestrate directed cell migration. So could you please elaborate on the importance of studying signaling pathways like those?
For sure. So both Wnt and netrins are highly conserved, and they are highly conserved both structurally and functionally. So they carry out the same functions in simple organisms like C. elegans, which is a roundworm, or in Drosophila, which is a fruit fly, just like they would carry them in humans. And so because they're highly conserved and because they play key roles in a variety of very important physiological processes, I decided to use C. elegans to study them, because in C. elegans, instead of having several family members of those Wnt, they have fewer. And so it's easier to, say, knock out one Wnt and see, or several Wnt and see a phenotype. And so to study these complex signaling pathways, you really need to, scale down the system you're using. And both those signaling pathways were involved in polarity formation and also involved in cancer, which were two things that I was studying and interested in. And basically, more specifically to the work that I was carrying out, they are both involved in patterning along the two main body axes. So the anterior posterior axis that goes from head to tail, and the dorsal-ventral axis that goes from back to belly. And so, I wanted to get an idea of how these two pathways are communicating with one another, because when cells or exons, which are the extensions of neuronal cells, are migrating and navigating, they are not exposed to one cue, they are exposed to a variety of cues altogether. And this information has to be integrated in a way and then interpreted to a defined outcome. So I was very interested to know what can then regulate the migration of a cell on one axis, and in particular, what would then regulate the transition from one axis to another, or the direction along that axis. So, so these are the type of questions that I wanted to ask. And then basically, if you study how these two signaling pathways interact with one another, then it can also give you an idea of how other signaling pathways are interacting with one another. If you can identify the cross-link between them, then it's normally not very specific to one signaling pathway, so it can shed light on pathway integration in general.
So I have a follow-up question to that. So did you know when you started your research that the Wnt and the netrin pathways had cross-links to other pathways? Like, were you aware of those?
So originally, when I came to Toronto, I came to study the netrin signaling pathway. So I wanted to get more information about what are the mediators of dorsoventral polarity, and so on and so forth. But I was working on I chose a very interesting organ, and that's the C. elegans gonad. And you have two cells that shape that gonad, and they migrate first on the anterior posterior axis. So they start from the mid body, and then they migrate to either the head and the tail, and then they switch polarity in 90 degrees and start migrating from the ventral to the dorsal side, and then they switch polarity back again and return to the mid body. So then I didn't have only dorsoventral guidance, I had anterior posterior guidance and dorsoventral guidance. So I was trying to figure out, how it all works together to kind of see the entire picture. And I don't remember if I mentioned it or not, but I didn't mention the genetic screens. So I started doing some genetic screens, and I realized, hey, there could be an interaction here. Why don't I figure it out? And it was a great journey. It was very, very fun. Yeah, that is that.
No, that's great to hear. So it just kind of extended into this bigger, more immersive project, I guess.
Yes. You always want to strive to get the bigger picture. You always want, because signaling pathways do not work in a vacuum. They don't work in isolation. Everything integrates with everything. So it's neat to see how it is all orchestrated.
Yeah. But a lot of our audience actually is not familiar with how genetic research is conducted. So could you tell us a little bit more about how you were able to conduct it, like the nitty gritty, such as like the type of equipment you needed or the specific techniques that we might even be familiar with, like from our studies, and even like the time this type of research might take?
Right. So first of all, I would start with the end of the question. So the answer to this question about the time is very different now than it used to be several years ago. So with the discovery of CRISPR, manipulating genes and genomes became really much simpler, much less time consuming, and much more directed and precise. So all this research that used to take years before can be done in weeks nowadays. So that's about in terms of the time frame. And basically, how do you go about studying a particular signaling pathway? You do that by looking at strains that would not express components of these signaling pathway mediators. So it could be would not express the ligand, or would not express the receptor, or would not express any downstream mediators, or alternatively, it could be any changes in the expression of these signaling components. So it could be either that they are their expression is reduced, or they are overexpressed. So you take those strains and you analyze the phenotypes. And not only you would analyze the phenotypes, you can also try to screen for other genes that might interact with them. And so that's the way you go about it. And so basically before, scientists were limited to existing strains, to strains that they were generating by random mutagenesis. And nowadays you can generate exactly the strain you want with the mutation in a particular base pair that you want. And it makes the process much cleaner and much faster. And in terms of the equipment, of course, microscopy is key. So you would need a light microscope, you would need a fluorescent microscope, because we tend to use a lot of fluorescent reporters to follow gene expression and to figure out a variety of, to look at, to try to map the expression of different proteins in the cell, and in this way try to figure out whether they are interacting with one another or how they can affect the expression of one another. So a fluorescent microscope, and also confocal microscopy. So then you can get a higher level of resolution and carry out also some live imaging as well. And in addition to that, most of you probably heard about PCR. So we use a lot of PCR for genotyping. We use DNA and RNA sequencing. Now it's quite instrumental as well. And of course, we use a variety of techniques, a variety of molecular biology techniques. So, you know, anywhere, anything really that you would use normally in molecular biology.
So I just, before I go to the next question, I just will point out how interesting it is that your research involves a lot of the things we learned in Bio 230, HMP 265, and Bio 130. I think Hawwa agrees with me. Yeah. And our audience, it should be interesting to our audience that the things we learned are actually applied in the research world.
Oh, absolutely, absolutely. Yeah. We don't teach you stuff that you don't need.
So
we try to make it useful.
Yeah. So my next question is, you have also studied how the Wnt and the netrin pathways can affect tissue patterning and neuronal connection in C. elegans. This was from another paper. So can you please explain how these findings could potentially be applied in the human context, or how these studies may influence our understanding of cell migration or cell polarity?
Exactly. So as I mentioned, those signaling pathways are highly conserved, and they play the same roles in C. elegans. And so any information, they play the same roles in humans. So any information you can glean from studies in C. elegans or in Drosophila are applicable to humans as well. And as a matter of fact, netrin was first identified in C. elegans, and it facilitated the identification of that cue in mammalian systems. So, and it facilitated the identification of the receptors and downstream mediators. So, you know, it's not only in theory, it's in practice that you can apply that knowledge to humans. So, first of all, this would be, so all the information that is gleaned in terms of how these pathways interact, and what's specific to this work was identifying that link between the two pathways, that MAM5, which is a Wnt receptor, being a regulator, a negative regulator of the netrin receptor, provided that connection between the two signaling pathways and provided an explanation of how you can get that 90 degrees turn, how the cell can stop migrating on one axis and start migrating on the other. Because basically, if you would have input from both signaling pathways, you would expect the cell to migrate diagonally, because it would kind of feed information from both. And so first of all, you can see how this information can be useful in terms of cell migration and in exon guidance. But also, these two signaling pathways play key roles in cancer as well. And of course, they would, because they do affect migration. But it's not only because of the migration. And so the information that can be gleaned from such studies can be very instrumental in helping us design different therapeutic approaches to a variety of cancers. So these are two ways in which you could use that information and apply it to the clinic even.
Yeah, sure.
Sorry, I just had a quick
I think
follow-up.
yeah.
Yeah, I was just wondering, like, do you know what types of cancers that might be like specific cancers that might help use, like that could be used as therapeutic?
Yes, absolutely. So for one example can be glioblastomas. So Winds play a key role in glioblastomas, and you can use the synergistic effect between the two signaling pathways as a way to treat glioblastomas, for example.
Sure. So I guess I can go now. So going back to the idea of, working with things that we learned in our courses, you worked with C. elegans, which is a model organism we hear a lot about in our courses. I think the first time we heard about it was in volume 130. So can you please provide some insight on the challenges involved with working with these model organisms, specifically C. elegans?
Right. So, basically, I am a fan of C. elegans. I don't see many challenges that are involved with using that organism. Quite contrary, as a matter of fact, because it's so small, because it has a small number of cells, this facilitated the identification of each and every cell in the body of the worm, and it facilitated the mapping of the entire nervous system of the worm and the different, synapses and the way those neurons connect. So it's huge. It's also transparent, and so you can carry out all your studies in vivo. The only disadvantage maybe can be because it has a small number of cells. So for example, when you want to work, nowadays we have the technology of analyzing single cells. So when you want to do that type of an analysis on single cells, then it becomes much more of a challenge. But other than that, it's a great model organism to work with.
So would that be, I guess this is going in another direction, but do you think that is also part of the reason why we have simulations such as open worm, which is specific to C. elegans and not a different model organism?
Yes, of course, there were a lot of things that were possible to do in the worm that were not possible to do in any other organism. So this is not to say that C. elegans is the only transparent organism. You probably know that zebrafish, the early embryonic stages are also so that transparency is very, very useful. And yes, C. elegans was the first one that the entire nervous system was mapped, which is huge. It's huge in terms of getting an idea of how these different neurons work together. And that was even before optogenetics. So now with the tools that we have with optogenetics, we have other ways of tackling it, but this was even prior to developing more genetic tools that are very powerful and useful.
That's great to hear. I know Mohammad works with C. elegans, so he's got his own opinion.
So which lab are you working at?
I work at the Gen Lab in the
Oh,
of molecular genetics.
okay.
Yeah.
Okay.
But I do more computational work, so it's a different sort of challenges.
Yeah. I know that May was trying to do a lot of single cell analysis. I know it could be a challenge.
Yeah, so it's interesting, I guess, that you mentioned C. elegans being small is a good thing, whereas with what I do, C. elegans being small is one of the challenges. Because I'm trying to identify C. elegans under the microscope using computer vision and machine learning.
Yeah.
And having a very small size, it's hard to extract features.
Right.
So.
Yeah, so I'm not a computational biologist, so.
But you have done
I can see the challenge.
Yeah, you have done a lot of fascinating research, but you've actually transitioned from research to the teaching and more classical and applied genetics courses. So what actually prompted you to make that transition?
That's an excellent question. So first of all, I really enjoyed teaching and mentoring. It's really a passion of mine. And I would like to spread the love of science. So you are the future. And this is my way of investing in the future. So I would like to help you get the right foundation and acquire the knowledge so you can go and do great things with it. That's my investment in the next generation.
That's really wonderful to hear. But you're actually
And
really
I should also add, especially in the field of genetics, because it's such an exciting time for the field of genetics now. There is a true opportunity to make a difference, really a true opportunity to make a difference. So, hey, go and do it.
But did you hear that? Genetics, not health and disease.
Exactly.
Well, I mean, whether you're a HMB major or you're a molecular gen major or you're an LMP major, most people have to take HMB 265. And a lot of students actually find it really challenging. And I guess it's because the content is quite new. It's a pretty intense course, I think you could say as well. Do you have any advice for students who are going to be taking this course or actually wishing to pursue further genetics courses who've already taken HMB 265 and are a little concerned about what the future holds?
Okay, all right. So first of all, definitely. So in HMB 265 in particular, practice makes perfect. You just have to practice as many questions as possible. It really helps get a better understanding of the concepts. So this is one suggestion. I also have to say that another key component is asking the questions and making sure that you understand the concepts. And in this course, there is a ton of support. There is really a ton of support. The profs are there to help you. The TAs are there to help you. All you need to do is reach out and ask. So this is the time to ask, not after you graduate. So you might as well use that support and ask and figure it out and get a good grasp of everything, because this is the foundation. This is really the foundation. And I should also say that there are many facets to genetics, and they're not all portrayed in HMB 265. So don't equate genetics to HMB 265. For those that are interested, I highly encourage you to take other genetic courses that focus more on the applications and all the current tools that can lead us to breakthroughs in the clinic. And this is really a very exciting time, to be involved in that. So I know that HMB 265 discourages a lot of people, and I really lament about that. Don't be discouraged from HMB 265. And also HMB 265, and you may have heard me say that when you took the course, it really selects out people that don't pay close attention to the details. And I don't think that should happen. And I don't pay close attention to the details as well. And I do tend to read a question and miss, 5 things that were mentioned. Don't be discouraged by that, because it doesn't mean that you cannot do well in the field.
And I mean, I do want to say when we took it in the fall, and I mean, we took it online, and I think we were all a little bit worried on top of the fact that it was like we'd heard all these horror stories, and then on top of that, it was online. But I do want to say, like, there was a lot of support. Like, that's definitely, like, just you responding to discussion board questions was like just the best thing, because we could make sure, like, okay, all the concepts are clarified. You taking the time to do extra office hours, like, it really helped. And I do want to say thank you for that, because
You're welcome.
the time was different and it was a big adjustment. So I think just the fact of having a little bit more support in such a big course too was great.
Okay, that's wonderful to hear. So yes, for all the new students, there is a ton of support. So use it, wrap your heads around it, and come to the genetic stream.
I also want to raise a point about HMB 265 is that most students often worry, we tend to worry about our grades and how, tests are going to be, so on and so forth. But I think one unique thing about HMB 265 is that you can genuinely feel how both Dr. Levy-Strumpf and Dr. Chang are very interested about what they're teaching. And, you can, I guess during the lecture, you can forget about your grade and test and everything and just enjoy the process of learning and excitement that there is in that course. I think this is something people miss when they take HMB 265. They worry too much about their grades.
Right. And I should say, and maybe you've heard me say that before, I should also say that, the grades might be important for you to get to wherever you need to get, or get your scholarships, or get to the programs that you are interested in. But they're not going to help you after you graduate. Nobody looks at your grades. They would look at what you bring to the table. They would look at your ability to problem solve. They would look at your, if you step up, if you are, if you're contributing or not. And these are the kind of soft skills that you need to acquire when you are in university. And they are more important than your grades. Really, I'm signing off that and mean every word of it. And I should say, quite contrary, like if you come with amazing grades and you don't have anything to back it up, it's even more disappointing. It's even more disappointing. So, focus on the learning. This is the most important thing. There are people that self-learn and do amazing, they don't have grades. So it's what you learn and what the skills you acquire that is far more important.
Yeah, for sure. So we're kind of coming to an end of our podcast. So our audience who have been following us for a while now, they know that it's time for our famous question which is the same for all of our guests. So who's your favorite scientist and why?
So that's the toughest question of all. It's very hard to pick a single scientist because there's so many that I like for different reasons. But if I have to pick one, then I'll pick Hans Clevers. I don't know if you heard about Hans Clevers. And I pick him because of the great science that is done in his lab and the innovative approaches that he takes. And it was the first lab that developed organoids. So these were intestinal organoids, and how organoids are now front and center in research, in brain research, in other research, in science and in preclinical trials. So if I have to pick, I'll pick him. Although, if you were to ask my HMB 431 students who is my favorite scientist, they would have probably picked David Liu because I feature many of his papers. And again, I feature many of his papers because of his innovation and the way he thinks out of the box. So very cool stuff. Yeah, these are, so Hans Clevers is my pick, and probably also for CRISPR-based tools and techniques, David Liu.
That's great. I mean, that's why we love asking this question, because we always get such a different answer from every prof. Right. And so we're learning about someone new. I'm sure if we can now Google Hans Clever, and we can learn a little bit more. Clevers, yeah. Sorry, and learn more a little bit about his research as well. We might actually link a little bit of a profile for him down below. So with that, we're really happy to talk to you, and we're really glad we got a chance to meet with you and talk about your research, and we'd like to end the podcast. Thank you again for joining us, and to our
pleasure.
To our audience, please stay tuned to our social media, and we'll be posting more, and you'll be hearing more from us in the next few weeks.
Wonderful.
And subscribe.
Yep, click subscribe.
Subscribe.